Good evening, dear students. So today we'll be talking about drug metabolism. So basically, what happens in metabolism? There is phase one and phase two. Phase one, we are converting your drug into your reactive metabolite, and then in phase two, we are further conjugating it and detoxifying it so that it is eliminated completely from our body. So basically, we are targeting to that is to make the compound inactive as well as to reduce the toxicity of the drug, and lastly to get excreted from the body. So drug metabolism, that is considers the metabolic change of drug and related organic compound, describes the metabolic process of various functional groups. So basically we are trying to change the functional groups, like if there is nitro we are converting into amino, if there is an methyl we are converting into carboxylic group. So these are the groups which are found in the therapeutic agents. We are converting it to another form, changing the functional group so that they get metabolized. So basically, these are the things that we are dealing, and this helps us to in understanding the basic drug interaction, how metabolism is related to drug interaction. So which drug has to is contraindicated with others, that can be done. So sometimes one of the drugs will be over metabolized or one will not be combination when you give it. So that might affect one or another. So these things are addressed in drug metabolism. So basically, we will be dealing with biotransformations with the help of specific enzymes. Okay, where specific functional groups are targeted. So this we are going to be covering in this particular topic. So the definition of drug metabolism is the process of converting drug into a product or inert substance after or before reaching to the site of it. So, what does it mean? Convert into a product means we are changing the form and converting into a new product, or we are converting into an inert substance so that it can be excreted from the body. So, metabolism is an essential pharmacokinetic process which renders lipophilic and non-polar compound to water soluble polar compound so that they are excreted from the body. Okay, so what do you mean by pharmacokinetics? Dynamics means what the drug does to the body, and kinetic means what your body does to the drug. So biotransformation is a specific term used for chemical transformation of xenobiotics in the living organisms. So xenobiotics are nothing but chemical substances that are not nutrients to the body, but from which comes from foreign body, which is not synthesized in basically which is not synthesized within your body, which comes out. Whatever food we intake, whatever medicines, they are all xenobiotes, which enter the body through ingestion, inhalation, or dermal exposure. Even the creams that we apply, so they also have to be entered. Okay. So this is about bio drug metabolism. So as we know that most of the drugs which we take are lipophilic in nature. To be absorbed, they must transfer through the lipoprotein membrane of the luminal wall of your GI, gastrointestinal tract. So that is the most important. So if it is too polar, then it will not get absorbed, it will be excreted as such. So in order to the drug to penetrate through the GI luminal membrane, lipoprotein, they have to be lipophilic. And then once in the bloodstream, these molecules can diffuse passively through the membrane. Passive means across the concentration gradient and distributed effectively to each and various target organs to exert their pharmacology. So once they get into the bloodstream, then they go to the specific region and show the effect. Because of reabsorption in the renal tubules, lipophilic are not excreted as substantial extent in the urine. So as we observed that if you want the drug to be excreted from our body, it should not be lipophilic. Otherwise it does not get filtered through the renal. So in order to get excreted, these compounds, which the drugs which you have taken, has to be metabolized into a polar form, and this happens during biotransformation. So the xenobiotics that meet their metabolic fate through various enzymes. So there are various enzymes, as we know, esterase, cytochrome P50. There are various enzymes which are present, which convert the change or convert the parent compound to its more water soluble, that is lipophilic, make it more lipophilic. Okay, once the metabolite is sufficiently soluble, it can be excreted from the sufficiently soluble. That is the most important. So that's why there is two terms, that is phase 1 and phase 2. Phase 1 is converting into a more polar compound, but once this cannot be excreted as such. So they have to be converted into hydrophilic. So that happens when you get in a conjugation where your phase 2 metabolism comes. So we have to understand that, and we should know the ADME problem, that is absorption, distribution, metabolism and excretion principle, which is used for determining the drug regime. So these are how. So all these parameters in, so absorption, distribution. If your drug is not absorbed or distributed, it is, if it is not absorbed, then it is excreted as such. If it is not distributed, it will not go to the organs. If it is not metabolized, it is not getting converted in a polar form, and if it is not getting metabolized, it is not getting excreted. So all these parameters are important. So if lipophilic drugs were not metabolized to polar readily water soluble products, they would remain inside our body and continuously show their biological effect, that is elicits their biological effect. So continuously, if you take one drug once, that means it will be available throughout your lifetime. That is dangerous. That is dangerous. Thus, formation of water-soluble metabolite not only enhance drug element but also leads to the compound that are generally physiologically, pharmacologically inactive and relatively less toxic. So this is overall how what happens to the blood, to the drug. So drug enters through various either through passive or active mechanism. The free drugs goes into circulation. The free drug might be bound to the protein, okay, in the tissues, or if the free drug is not metabolized, unchanged drug is eliminated. If it is polar, it is eliminated through the kidney. If the free drug is not polar, it can be converted into its metabolite in the liver and then excreted through the kidney. Then, so this is your basic thing, that is absorption, distribution, metabolism, and so it starts with absorption, distribution, metabolism, and elimination. So as we said that in drug metabolism, traditionally it considers detoxification process, means you reduce the toxicity. Unfortunately, it is incorrect to assume that drug metabolic reaction is always detoxifying. There is no gold standard that once the drug gets converted into an inactive or detoxified product. So that's not a classified this. But it helps in detoxification. Many drugs are biotransformed to physiologically active metabolite. Yes, once you are taken, as such it is inactive, but once it enters in our body in vivo, it gets converted to an active form. That will show pharmacological and toxic once it gets ascribed effects ascribed to the parent drugs. So occasionally the parent compound is inactive administered but metabolically converted to a biologically active metabolite. This type of compounds are known as prodrug. As such, they are inactive, but once they enter the body, they get converted to an active, pharmacologically active in comparison to the parent. So why do you want biotransformation? So the biotransformation process converts your non-polar lipophilic compound, that is lipid soluble compounds, which the body cannot excrete, into a more polar hydrophilic compound, that is water, which the body can excrete in a short period of time. So basically your biotransformation is for removing the drug from the body to convert into non-polar to polar compound. Because if the water soluble non-polar compounds are not metabolized to polar compounds, they remain in the blood and tissue and maintain the form for a long indefinite time, which is not good. So we can classify these metabolites into four categories. One, your drug gets converted into an inactive metabolite, which is good. Second is the metabolite retains its activity. Third is metabolite with different activity. You start with one activity, when it gets metabolized, you get a different active compound. And third is bioactivated metabolites, that is pro-drug technique. So these are the four aspects of metabolites. Let's start with one, that is inactive metabolite. Some metabolites are inactive, that is their pharmacologically active compounds become inactive. That is hydrolysis of protein to para-aminobenzoic acid and diethyl ethanol amine results in loss of anesthetic activity of protein. So this is one of the examples. Second is oxidation of your anti-cancer drug 6-mercaptopurine to 6-mercaptoric acid results in loss of anti-cancer activity. 6-mercaptopurine gets converted into 6-mercaptoric acid. Mercaptoric acid. So this was the first technique converting inactive compound. Next is retain similar activity. Now let us say codeine, which you have studied. It is methylated morphine. So once this gets demethylated, you get a more active analgesic morphine. So this is one example where some metabolites retain the pharmacological activity of the parent compounds to be greater or lesser degree. Next is phenacetin, which it's metabolized to paracetamol. So that is acetyl O, which is acetylated, gets deacylated to an active paracetamol. So this is one of. Next example is imipran, which is when demethylated, you get an ecoactive antidepressant desipramine. So these all compounds retain their activity or show better activity than the parent compounds, which is metabolized. Next is you start with one compound, that is metabolite. Your parent drug has one activity. Once it gets metabolized, it gets converted into another compound, active compound. Let's give an example of isoproside, that is isopropyl derivative of nicotinic acetam. Gets dealkylated to isoniazide, which is an antitubercular acid. Next is your retinoic acid, that is vitamin A, gets isomerized to isoretinoic acid, which is an anti-active. So this is an example where some model develop different activity than the parent compound. The next class is about bioactive metabolite, that is prodrug, that is activation of inactive drugs into its active form. Okay, these compounds are called as prodrugs. Prodrugs are more stable, have better bioavailability and less side effects and toxicity. Examples: levodopa. Levodopa is carboxylated form of dopamine. So it gets decarboxylated in the neurons to active dopamine. So these are used as anti-Parkinson. So levodopa can't as such penetrate into the dopamine, penetrate into the brain. So it has to be decarboxylated. Once it gets decarboxylated, its lipophilicity increases and it enters the brain to show its effect. Next example is salindac. Salindac is nothing but a sulfone derivative, sulfoxide derivative, which is reduced to its active sulfide form, that is S form. Sulfoxide derivatives is more polar, so distribution is so this gets reduced by the intestinal lumen to your sulfide molecule, which will show the necessary anti-inflammatory agent. Whereas your sulindac as such is sulfoxide form is inactive. Next is benorelated, that is combination of your aspirin and paracetamol. So your aspirin has your carboxylic group free and your paracetamol has an OH group at the para position. So they two will combine to form a ester moiety, and together the combination of that is known as benorelate. It is known as benorelate. It is known as benorelate. So this is known as mutual prodrug. Other example is enalapril, which is hydrolyzed to enalaprate, which is a potent antigen. So this is an ester. So when it gets hydrolyzed, and you get your potent enalaprate. Ester gets converted to enalaprate, which is your potent anti-hypertensive agent. So in turn, the process is the drug gets metabolized into more polar substance, and the most important target is your liver, which metabolized, and the inactive form is excreted either through urine or through bile. Okay, so this is your oral blood method. So as you see this chart, which shows that highly lipophilic drugs gets accumulated in the body. So they are slowly converted into a more polar compound by phase 1 metabolism. These polar compounds are then further metabolized using phase 2 method, that is glucuronidase, converted into hydrophilic group compounds and excreted. Lipophilic directly through phase 1 metabolism, polar phase 2, and hydrolytic. Polar compounds are directly metabolized by phase 2 mechanism, that is glucuronidase, that is conjugation and excreted. And hydrophilic compounds are excreted as such. So let's say some of the general processes which occur during your phase 1 and phase 2 metabolic pathway. So in phase 1 there is nothing, that is a functionalization reaction. So one functional group gets converted into another. So in doing so they get converted into your polar compound. So some of the reactions that are involved in phase 1 reactions are oxidation reaction, reduction reaction, hydrolytic reaction. And some of the reaction in phase 2 or conjugation reaction are glucuronic acid conjugation, sulfate conjugation, conjugation with glycine, glutamine and other amino acids, glutathione or mercaptouric conjugation, acetylation and methylation. So these are some of the processes which are involved in phase 1 and phase 2 biotransformation. So this is the overall representation which reactions happen: oxidation of benzylic, allylic, alicyclic carbon, carbon heterocyclic system like CN dealkylation, CO dealkylation, S dealkylation, S oxidation, desulfurization. So the double bond, SS double bond formation. Reduction is conversion of which all groups: aldehydes, ketones, nitroazo compounds are in. Hydrolytic reaction involves ester, amides, epoxide, arene oxide by epoxide hydrolysis. These are the reactions that are involved. And phase 2 conjugation drug metabolic involves conjugation with glucuronic acid, sulfates, glycine, other amino acids, glutathione, mercaptouric acetylation and methylation. So overall this is what happens in phase 1 and phase 2. Most importantly, where these things happen, the sites for drug metabolism. So which all tissues other than kidney, other than liver? Kidney, lungs, adenoid, placenta, brain and skin. So these are the sites where your biotransformation occurs. And liver is the most important organ for your drug metabolism and detoxification of endogenous means which is present inside the neurotransmitters and all that, and exogenous compound that are xenobiotics. So they are the targets. So liver is a. Others is intestinal and mucous, especially for oral. Like if you see some of the drugs, they get metabolized in your liver, intestinal mucosa. Intestinal mucosa. Orally administered drugs that are absorbed into the bloodstream through the GI must pass through the liver before they are distributed into the body component. Therefore, they are susceptible to hepatic, known as first pass effect, before reaching the system. Depending upon the drugs, the metabolism can sometimes be quite sufficiently and result in decreased oral viability. So what happens when they are metabolized in the liver? That will reduce the viability of the compound. So this is the side effects of your hepatic metabolism, because if they go into that, they will undergo first pass effect and make the drug inactive. So these are some of the sites where the drug metabolism occurs, enzymes responsible, and what is the action. So enzyme esterase and lipase, which are present in your intestine, they are involved in carrying out hydrolysis of many esters, basically in your prodrug approach. Bacterial flora enzymes, which are associated with bacterial, they are present in intestine and colon. They play an important role in reduction of many aromatic azo and nitro compounds. So they are present in the bacterial flora of intestine and colon. Glucuronidase enzymes, which are present in your intestine, they hydrolyze glucuronic acid conjugate excreted in the bile, thereby liberating the feed drug and its metabolite for possible reabsorption. So as we know that during glucuronide it gets in. So this can be again got back into the circulation using glucuronidase enzyme, which are present in your intestine. What's the next step? Now let's go one by one. Oxidative reaction, that is phase 1. So there are two types: microsomal enzymes, which are responsible oxidation reaction, and another is non-microsomal enzyme. So microsomal enzymes are basically found in the smooth endoplasmic reticulum of your lysosomes, and the enzymes involved are cytochrome 450 monooxygenase, flavin monooxygenase. These are involved. Where non-microsomal enzymes are found in cytoplasm and mitochondria of liver cells. Examples of aldehyde dehydrogenase, aldehyde and alcohol dehyde, and monomer. So these are, as we have studied, that these are involved in the hydrolysis, in the metabolism of your neurotransmitters like your epinephrine, norepinephrine. So you know where is the site. This is found in cytoplasm and mitochondria of liver cells. Now let us go to the first enzyme which is involved in your oxidative biotransformation. That is cytochrome 450 monooxygenase in your oxidative biotransformation. The enzyme cytochrome. So of the various phase 1 reaction, oxidative biotransformation process is the most common and important in drug metabolism. As you know, which is the most abundant process that occurs in the body, it is oxidative biotransformation. Basically what happens, the drug that is present, RH, which is involved, gets oxidized into your metabolite, that is O gets inserted inside your synovium by the following reaction. So you got an substrate, that is RH, NADPH, which is a cofactor, and in presence of oxygen and acidic medium gets converted into ROH, NADPH plus, and water. So enzyme system carrying out this function are referred as mixed functional oxidase or monooxidase. So they introduce one OH into your substrate. Okay, they use they require molecular and reducing at that NADPH. So in the oxidate, one atom of oxygen gets introduced into your substrate, that is RH gets converted into OH, and another oxygen gets transformed into your water. Okay, so this is what is happening. The mixed functional oxidase system is actually made up of several components, the most important being the superfamily cytochrome enzyme, which are responsible for transferring an oxygen atom to the substrate. So cytochrome enzymes are basically what? Monooxygenase enzymes. Okay. The cytokine enzymes are heme proteins. Heme protein means they are, heme portion is an ion containing porphyrin called protophyrin ion, and the protein portion is called as aporose. Cytochrome is found in high concentration in liver and major organs involved in metabolism of them like liver, kidney. They are all present over there, lungs. The presence of this enzyme in many tissues like lungs shows that these tissues have drug oxidizing capacity. So if this particular enzyme is present in that particular, so most of the xenobites are metabolized by oxidative process. So why that name cytochrome P450? CYP P450? Why that P450 has come? So during this reaction, when it happens, the reduced Fe+2 forms of this enzyme binds with the carbon monoxide to form a complex which is distinguished spectroscopically absorption maximum at 4. So this complex between Fe+2, that is ferrous ion, and your carbon monoxide of the enzyme gives a sharp absorption at P450. That's why it is known as cytochrome. It is one of the most important feature of this hepatic mixed function oxidase system is its ability to metabolize almost unlimited number of diverse substrate by various oxidative transfer. So it is this particular enzyme is a substrate for many of the target substrates. And cyclochrome monooxygenase are located in the endoplasmic reticulum, smooth endoplasmic reticulum, and are highly organized and complex network of intestinal system and particularly abundant tissues such as lung. And this is involved also in the biosynthesis of many steroidal hormones and metabolism of certain vitamins. So this is the overall function. So if you see this particular chart, if you see the top, it is cytochrome P50, it is present as Fe+3 form. When the substrate gets added, you get cytochrome P450, Fe plus, and RH gets incorporated into your enzyme. Then comes your NADPH. So NADPH is a reducing agent, so it incorporates one oxygen into it. In present cytoplasmic reductase, so it introduces one electron into the system. Moment it introduces one electron into the system, your Fe+3 of cytochrome P450 gets converted into Fe+2 form. And this Fe, some part of this cytochrome P50 Fe+2 combines with carbon monoxide which is present in our blood, and it forms a complex, that is CO cytochrome P450, that is reduced form, along with gives an chromophoric absorption at 450 nano. Okay, so this is one thing that is happening. When it combines with the, you get this. So when this combines with oxygen, so till now there was no oxygen. Once oxygen gets incorporated into this system, that is oxygen gets added to this system, you get the next form. So it is reduced form, oxygen gets introduced. This reduced form, the oxygen gets introduced. Now again the NADPH will give one electron in presence of cytochrome P450 reductase. Your O2 gets activated, gets converted into an active form. You can see it, it gets converted to an active form. And at this stage, it will react with two molecules of two parts of proton H plus to form water. So here the first oxygen is removed and it reacts with two protons to form water. And this activated oxygen species now that gets introduced into your substrate and gets removed, and you get back your Fe+3. So in this case you get your Fe+3 form. This is how the cycle continues. This is how the cycle continues. Now let's name the naming of the cytochrome. The gene encoding cytochrome CYP enzymes and the enzymes themselves are designated by the abbreviation CYP. So the genes as well as the enzymes are designated as CYP, which is followed by an Arabic name indicating the gene family. So here the 2 represents the gene family and capital red indicating the subfamily and another numerical number for individual. So each has the parent name. CYP is the common name of the gene of the enzyme, followed by an Arabic number that is indicating the gene family, followed by an capital letter that represents the subgene of the family, and the number which is representing the individual gene for that particular enzyme. So cytochrome P2E means is the gene encoding cytochrome 2PE1 and one enzyme involved in the paracetamol. So these are some of the examples of cytochrome, that is cytochrome 1A2, cytochrome 2D6. So these are some of the enzymes, the genes which they are involved are given in the particular form. So this is about your isoforms of cytochrome P4. And this particular enzyme is involved in various reactions. Your benzene gets converted into your iron oxide, which gets converted to phenol and further oxidized. Benzylic allylic compounds gets converted into your phenols. Double bonds gets converted into epoxide. Okay. So this various disulfurine gets converted into your SH, bond breaking happens, and hydroxylation happens, and dealkylation, oxidative deamination, and oxide formation. These are various reactions in which your cytochrome P50 family are involved. Cytochrome P50 family are involved. So the next, this thing is reductive reaction. So here basically reduction is involved is carbonyl, azo compounds and nitro compounds. Basic things are involved. So what happens when carbonyl aldehydes get reduced? You get a carbonyl alcohol. Nitro and azo compound leads to amino derivative. The hydroxyl and amino moieties of these metabolites are much more susceptible to conjugation because it becomes more polar and they can conjugate with various conjugating agents like glucuronic acid, sulfates, acetates, and they can be eliminated from the body. So these are the various things. So reduction of aldehyde ketones to carbonyl. Aldehyde gets converted into primary, ketones gets converted into secondary alcohols. So which mostly the enzyme aldoketo reductase are involved in the reduction of aldehyde and ketone. So aldoketo reductase carry out the bio reductions of aldehydes and they are found in livers and other tissues like kidney. Oxidoreductase enzymes are involved. So what does it mean? This particular enzyme, depending upon the cofactor with which it is associated, they can undergo either oxidation or reduction process. Okay, of the, so by oxidation, both oxidation and reduction of your reaction can also reduce aldehydes and ketone. For example, liver aldehyde dehydrogenase is an NAD plus dependent oxidoreductase that oxidize ethanol and other aliphatic alcohol into aldehyde ketone. So this is oxidation reaction. But in the presence of NADH, that is a reducing agent, however, the same enzyme now can reduce your carbonyl derivative into your corresponding alcohol. So the dual nature of oxidation and reduction. So you can take the example of chloral hydrate. There is lots of water molecule. Okay, so that gets converted into chloral, which is further reduced to your trichloroethanol, which can be further transformed by phase 2 biotransformation by glucuronidation. Okay, to an inactive conjugate and gets readily excreted. So this is the way the drug can be eliminated from the body. Other example is acetophenone gets oxidized to your S-methyl form and another form. So next example oxysuran. So it is a carbonyl derivative. Okay. This gets reduced to your OH, that is oxysurinol, and after a pharmaca and contributes substantial to the immunosuppressive effect of parental. So this is the example of activation. So reduction of oxysurin. Studies indicate that oxysurin has greater immunosuppressive effect in those species that form alcohol as a major metabolite. So this is an example where metabolite is also active, that is oxysurin. This happens by reduction of your carbonyl compound in your oxysurin to your OH, which also has immunosuppressive effect and thus increasing the effectiveness of your parent compound. Other example is reduction of nitro and azo compound to your amino metabolite. This reaction are carried out basically in the presence of NADPH dependent microsomal soluble nitro reductase present in your rival. Microsomal means it is present in your endoplasmic reticulum. A multi-component hepatic microsomal enzyme requires NADPH appears to be responsible for your azo reduction. So this NADPH are all nothing but your cofactors which are essential for your activity of your parent enzyme. Okay, in addition, bacterial reductase present in here can also reduce nitro and azo compounds, especially those which are absorbed poorly or excreted mainly in the bile. So these are some of the reactions of nitro and other azo compounds. So nitro gets converted to nitroso and hydroxyamine and amine. This is the sequence. Okay. In case of clonazepam and nitrazepam, this gets converted into 7 amino metabolite derivative. Azo compounds gets converted to hydroxy derivative, which further gets converted into your amine. Okay. So these are sulfapyridine. If you take sulfasalazine, gets converted by azo reductase to its corresponding sulfapyridine and 5-amino salicylic acid. So these are some of the reactions involving reduction. So miscellaneous reduction, like you got salindac, your sulfone gets converted into sulfite, which shows its anti-inflammatory action. Disulfiram is a disulfide linkage. The bond breaks, you get SH molecule, that is NN diethyl thiocarbonic acid. So which will show its effect. Okay. So next is hydrolytic. What is hydrolytic reaction? Hydrolytic reaction is basically involved with esters and amides. Esters and amides. So these enzymes convert your esters to and amides to your alcohols or acids. Alcohols or acids which are more susceptible to conjugation and excretion than the parent esters and amide. The hydrolysis is a major biotransformation pathway containing ester function. Classic example of ester hydrolysis is metabolic conversion of aspirin, that is acetyl salicylic acid to salicylic acid. So this is one of the classic which is study. What's the next thing? Cocaine. So cocaine has many things. Okay, cocaine has N-methyl. There is one ester moiety, that is acetate, and your benzoyl ester. Okay, the methyl group is hydrolyzed methyl ester and then benzoyl ester. The first thing happens is your hydrolysis of methyl ester, you get benzoyl ecogenine, and when the hydrolysis of your benzoyl group occurs, you will get methyl ecogenine. Okay, so these are the two products happening. So what is the happening? Basically hydrolysis of cocaine to give methyl ecogenine, however, and also occurs in plasma form as to a major extent in your blood. So this is what happens to your cocaine once it goes inside the blood. Other examples of this thing is hydrolysis of amides. So you have procainamide, so there is an amide bond and there is a diethyl amide. Whereas in case of procaine it is an ester, in procainamide it is an amide. So bonding, your breaking of amide bond is slower than that of your amine bond. No, sorry, your ester bond. So this ester bond gets easily broken to your para-aminobenzoic acid, and your diethanol amine. So this is your hydrolysis of ester and amide. So now comes your phase 2. Now comes your phase. So in phase 1, what you are converting? You are converting it to a mole polar form. Okay, non-polar, that is hydrophobic to polar compounds. So but this does not fully solve. So for further making it water soluble, it has to be conjugated with any polar compound. So here comes your phase 2 metabolism. So in phase 1, mechanical reaction are not produced sufficiently hydrophilic. So it is not sufficiently hydrophilic. It is just polar but not hydrophilic or inactive metabolite. The drug or its metabolite formed from phase 1 from the phase 1 reaction undergoes phase 2 reaction. These are capable of converting metabolite into polar and water soluble compound, and all these processes are high energy and enzymatic, and most of the enzymes that are involved are transferase, mainly found in liver and to the lesser extent in intestine and other tissues. So this phase 2 mechanism basically happens in your liver. So as we know that phase 1 is the functionalization reaction and do not always produce what? Hydrophilic metabolite. Okay. So they have to be converted, and phase 2 mechanism or conjugation reaction can convert these metabolite into more polar and water soluble form. Water soluble form. So many conjugated enzymes accomplish this object by attaching small or polar ionizable endogenous molecules such as glucuronic acid, sulfate, glycine, glutamine to the phase 1 metabolite or parent xenoid. The resulting conjugation products are relatively water soluble and readily excreted. The other phase layer, methylation and acetylation, do not increase the water solubility. Okay, that is one exception. So first one is glucuronide and conjugation. In glucuronidation, it's the most common pathway in the drug made. Why? Because glucose is readily available in our body. It gets oxidized into glucuronic acid, and numerous functional groups can combine with this enzymatically with the glucuronic acid. And glucuronide moiety, when attached to a xenobite substrate, greatly increase the water solubility of the conjugated. So that all favors your easy metabolism of your. So this is your basic structure. It's a glucose moiety which is attached to your uridine 5-phosphate, and at the sixth position your CH2OH of your glucose gets converted into your acid, that is known as glucuronic acid. Example is chloramphenicol. This is reacts with glucuronic diphosphate, uridine diphosphoglucuronic acid combines with a chloramphenicol that has an OH, forms an ester moiety, and your compound is okay. So here there is an OO linkage, that is glycosidic, or there is an acid. So different targets will react in different form. Next is sulfate. So sulfate is primarily used for metabolizing of your excretion of phenols, occasionally alcohols, amines, and anhydrous compound. In contrast to glucuronide, the amount of available sulfate is rather limited. Mostly anhydroxyl compounds are prone to sulfate conjugation. The body uses significant portion of sulfate to conjugate numerous endogenous substances like steroids, heparin, chondroitin, catecholamines, and thyroxine. So sulfate conjugation happens in the following way. Process involves activation of inorganic sulfate to it, the coenzyme 3-phosphate adenosine 5-phosphosal, that is PAPS. Okay, the sulfate reacts with your adenosine 3-phosphate to give your adenosine 5, that is AP, adenosine 5-phosphate, which further reacts with your another molecule of phosphate to form phospho 3 phospho adenosine 5 phosphosulfate. So fifth position sulfate phosphate also forms a complex with your sulfate, and the OH at the third position of your sugar will form a complex with your coordinate with your sulfate, together forming the PAPS, that is 3 phospho adenosine 5 phosphate sulfate. So this is your main intermediate which is involved. So this will form a complex, will be involved in the transfer of your sulfate to your acetaminophen. So acetaminophen can do two types of phase 2 mechanism, that is your glucuronidation and sulfate conjugate. So both the rings are favorable, but most one is your O-glucuronide is more favorable. Next is conjugation with glycine and glutamine. Both of them are amino acid. In case of glycine, your R is H, whereas in case of glutamine, the R is your propionamide. So these are the R functional group. So basically, when an acid reacts with ATP, it forms adenosine monophosphate, which in presence of your coenzyme A replaces your adenosine monophosphate and to form an acyl coenzyme A intermediate. So this intermediate of acid which is formed will act on this particular amino acid, that is glycine or glutamine, and in presence of an acyl transferase. So acyl group gets transferred into your glutamine to form an conjugate which can be excreted. So it can be, when R is H, it is glycine. When R is propionamide, it is glutamine. In this reaction, coenzyme A is released. Next is glutathione conjugation. So glutathione conjugation contains a mixture of glutamate, cysteine and glycine. So when the enzyme glutathione S transferase is present, the SH of your cysteine, the SH, that is thiol group of your cysteine, will combine with your any electrophile substrate which is present in the presence of enzyme glutathione transferase. So if there is an electrophile is what? Electron loving, that is electron deficient. So these thiols are electron rich, so they will form a bond with your electrophile and thus help in excretion of your compound, that is glutathione adduct complex is formed, which is then excreted from the body. And next example is acetylation, which is more prominent in primary amines, sulfonamides, hydrazines, hydroxides, and primary aliphatic amines. So these are the targets for your acetylation. The amide derivative formed from acetylation of the amino acid are generally inactive and non-toxic, and the primary functional group for acetylation is to terminate the pharmacological activity and detoxification of the compound. So this is what is the role of acetylation. So some of the example is aniline to acid, anilide, acetylation and sulfonamide into acetyl sulfonamide. So this is some of the example. And the last one is methylation. Last part of your this thing is methylation. So methylation generally does not lead to polar or a water soluble metabolite, except in some cases where there is a quaternary ammonium salt formation. Most methylated tend to be pharmacologically inactive. The coenzyme involved in methylation reaction are S-adenosine methionine. So methionine is an amino acid with an S-methyl group that forms a complex with your adenosine monophosphate. So when the substrate with RH, OH, NH2 or SH combine, So the methyl group of your methionine gets transferred to your substrate and you get methyl ether and methyl or thioether intermediate, and this reaction is catalyzed by methyl transferase enzyme. Other examples for methylation is COMT, which is an enzyme that is catechol O-methyl transferase, which converts your epinephrine, dopamine or non-epinephrine into your nor-meta-epinephrine, okay, or 3-methyl-tyramine, which are its metabolites. Okay. So some of the other examples are nicotine. Nicotine is nothing but your pyridine moiety at the third position. You are connected N-methyl pyrrolidine. So this gets your methyl gets introduced to your pyridine moiety and methylation, and it gets excreted. Next example is nicotinic acid gets converted into N-methyl, that is triglyceride, which is also excreted from the body. So these were some of the examples of various phase 1 and phase 2 mechanism. In summary, the reaction which happened, which enzyme is involved, and what are the functional. Like in case of glucuronidation, glucuronide transferase, OH carboxylic acid, NH2 and sulfonamides are used. Whereas sulfonation, sulfotransferase, OH, NH2 sulfonamides, NH, that is oximes are there. Hydroxyl amine is there. Glutathione and transferase, alkyl halides, alkyl nitrates, epoxide and lactones, which are electrophilic centers, means electron loving center, means they are positively charged. Okay, they are involved. Acetylation, mostly acetyl transfer, OH, sulfonamides, NH2, alkyl amines, hydrazines and all are involved. Amino acid class, that is glycine and glutamate, that is acyl transferase, CO group containing NH2 groups are involved in this. Methylation, methyl transferase, that is OH, NH2 and SH are